right. Our project title was the Nano Frontier, Cancer Detection. I'm Zeke. That's Brian. Sorry, I didn't mean to introduce you.
It hurts, man.
We were mentored by Dr. Christopher Collin. Sorry. Coughlin, my bad. All right. The need. Estimated cancer deaths in U.S. alone have been 585,720 in 2014. The majority of those consisted of lung, prostate, and breast cancers. The longer you go with cancer, meaning the more progressed you are in the development of the cancer in your cells, the more lethal it is. So early detection here will save lives. And as lethality increases, treatability decreases. So many TYs. I'm sorry, guys. Now, the research objective for our project was to make nanosensors that detect cancer biomarkers. And these biomarkers consist of proteins, antibody fragments, DNA and RNA fragments as bases for that. Now, these biomarkers, they scan your blood and they're used for early detection, as I said. They're taken really in pill form because anything in your body will leave, even blood cells, they'll leave your body, their circulatory system, everything gets cleaned out. And they're expected to be a week maximum that they can live in your body. They're taken in pill form, and then they're your first layer of defense for early cancer detection. Now these can be used by major hospitals and smaller clinics, anything that has a lab, really, including our FID tags. The two main components of these tags are the tag itself and the tag reader. Now, these are way more advanced compared to the barcode scanners that you have at your regular stores. Whereas the barcode scanners, you need a direct contact. Whereas the RFID tags, you can have it underneath skin, underneath anything, because it's based off of radio frequency. RFID stands for radio frequency identification. Now the RFID tags come in two types. There's your passive and your active. The passive RFID tags require activation before they can transmit data. There are two components, which is the antenna and the microchip, versus the active tags that have their own battery supply. Usually it is an actual battery, but sometimes they can be solar. Now in our case, we wouldn't be using solar energy because we'd be inside the system, in your system. So the active tags can transmit data without anything except the chip itself. And the way it would transmit, it would send out radio signals via antenna, and it receives power from the reader, meaning the battery. In this case, the reader picks up the signal and identifies a tag, and it can identify any type of cancer that it has been programmed to.
The idea with these tags is you would have them on the sensors flowing within the bloodstream, and the sensors would have some kind of, say, antigen that responds to the cancer biomarker. And when it detects it, it'll change the state and release the signal. And your reader will now pick up this signal, and you'll now know, oh, hey, something's been detected. We'll do this a bit later, but there are two ways we're thinking of to actually alert the reader. But we've thrown this word around, biomarkers, and they're organic substances in your blood that relate to certain biological processes. But they're not only restricted to cancers, they could also be just something natural, things growing, developing. So we have to make sure that we isolate the specific cancer antigens that relate to these cancerous symptoms. You can find them in most any bodily thing, like blood, tissue, urine. We're focusing on the blood because it's easy to get nanobots flowing in your blood system. We wouldn't want to put it, say, in the urinary tract. That's a bit more invasive and not something we're looking to do just yet. So these are some common blood-borne biomarkers. This first one, the prostate cancer. We have two main biomarkers, both of which are naturally occurring, but appear in elevated levels when you have prostate cancer. And so that's something we need to consider, the sensitivity of these sensors. Because if it's constantly going off, and you're constantly thinking you have cancer, obviously they're not doing their job. So that's something you have to consider. There are two types of lung cancer that we're looking at. The first one, carcinoembryonic. As the name might suggest, you can find them in embryos, but also in the lungs of heavy smokers. And so, as you might imagine, heavy smokers typically do develop lung cancer. The other one, lung small cell. This one, neuron-specific anaphylaxis. It's typically related to neuron-degenerative diseases, which lung cancer is one, but the other is dementia. And that's a rather heavy disease to come down with, but we could theoretically detect the early symptoms of dementia. This last one, breast cancer. We have two main antigens, 15-3 and 27-29. However, as it happens, 15-3 appears a later term than 27-29, stage 2 or 3, which at that point, early detection has probably failed you. So we're going to be focusing on 15-3. Okay. This technology is not only restricted to cancer detection. This is something that nanotechnologists have been looking at for years. And the biggest alternative that we've seen is the blood sugar monitor. These sensors, theoretically, could pick up and monitor your blood sugar levels. And so if you were diabetic and you no longer had to stick yourself with a needle each day just to get your reading, if you could just use a phone app and say, oh, this is my blood sugar for the day, it's a much better alternative. And the only price to pay is perhaps taking a pill once a week. And as I mentioned, you can also detect dementia. In certain infections, they also have certain biomarkers you can read. Genetic disorders as well. Some of them can be caught with these markers. Now these are some points of consideration, things where there might be a problem that we have to iron out or fix. Blood toxicity. In the end of the day, we are putting machines, foreign bodies, into the body. And as it happens, the body does not always appreciate this. The machines themselves could do you harm, or your immune system could have a negative response and perhaps kick them out or have some nasty side effects for the user. This is something we need to get figured out before we do anything inside the actual human body. The other one is the sensor size and the free flow in the bloodstream. These things are in your blood cells, and if either they are hindered by the blood cells or they hinder the flow of your blood, once again, they're doing more harm than good, and we need to make sure that they're both small enough to move without causing any restrictions or constricting the flow of blood. And as we've mentioned, the sensitivity, because if you have constant false readings, which is a pretty common thing actually in modern early cancer detections of false positives. Once again, it causes people to panic and they lose their trust in this kind of technology. You don't want them to not trust the things that are trying to help them. And now with the alerting the user, comes back to the RFID tags. One idea is to perhaps have the reader put in a wristband that you would wear on your wrist, and it would constantly be searching for these activated biomarkers, the sensors. And if it were to detect a change, it would light up and you would know, oh hey, something's there. Another idea is to perhaps put the reader inside a cell phone and work with an app. And so you would just do a scan and then you would see which marker has gone off, which cancer it has potentially detected, and maybe even a time frame to go to the doctor. This is a bit farther ahead and requires programming and a bunch of other fun stuff. The last bit, persistence versus refreshment. This goes to the pill we mentioned, because there are two schools of thought. Either you make the nanobots and you put them in your bloodstream and they stay there for long periods of time, in which case you have to find a way to keep the nanobots from leaving the bloodstream, because everything cycles out in time. And so either you keep them there and they just avoid the recycling process, or you constantly refresh your supply of these little machines, and that's where the pill comes in. That's probably the easiest way to do it, but it might be a bit less cost effective because now you're having to make a whole bunch of these things and constantly take them. But that's generally our idea. Any questions?
Okay, so you're,
Sure.
I'm sorry, go ahead, Jason.
Oh, he just wants the names.
Oh, okay. So is your part of the new detection method is the RFID, or that's already in place?
Oh, that's something that already exists. We're just going to use them with the sensors to correlate or work with the actual detecting of the cancer. Because the information has to go out, and you have to
Okay.
know it's picked up something.
So your contribution is the sensor that goes with the RFID,
Yes.
okay, that makes it what, faster, in terms of being able to transmit the information that what the RFID now are doing, or I still didn't get the actual
Well,
difference if you have a sensor or not a sensor, I
yeah,
guess.
well, the sensor itself is what picks up the biomarker. The RFID has nothing to do with that. It just tells you if the sensor itself has picked up something.
Okay.
Yeah, it's just our alert, how we alert the user.
Yeah, sure.
What material have you considered using as the sensor?
We haven't ironed that out yet, but there has been projects in which people have used nanobots for medical medicine delivery inside the body, and they had successful trials. They didn't have any problems with blood toxicity or anything. So we're gonna be looking into that, and I'll get back to you when we know exactly what material they use.
Alright, considering that the sensors are constantly searching for cancerous cells until they're replaced by new blood, they're going to be constantly using energy, so what energy source are you going to use?
The tags themselves are powered by the tag reader. For the sensor itself, we were thinking of putting some kind of antigen that responds to the biomarker, and so it's not actually like powered, it would just, most of the power would come from the RFID reader, but as far as knowing if this cancer's there, it would be when the antigen we put on the bot reacts to the antigen inside the body.
What's the biological response right now of the RFIDs on this certain types of cancer? The percentage of rejection and the percentage of actually successful operation inside the body?
We don't
Have
know,
you looked at that?
we don't know if anyone's actually done this in the body itself yet. The
Right, or
RFID
any
itself.
organism, it doesn't matter if it's human or if it's tested already. Has there been any report on that?
Well, RFIDs are used in dogs.
Yes,
for
so what about
tracking.
like in dogs? Was it successful in
There have
the
been,
rejection or?
I don't believe so. It's a rather common thing. I don't think it's too common. We haven't looked too much into it, so I could be entirely wrong, but I haven't heard anything of
Right.
dogs rejecting these tags.
Right, because if it's not, then, yeah, if you have 100%, acceptability in the bloodstream, then that is really good. Because that's like you said, the immune system is always going to be detecting anything foreign, and so that's where the problem that you will probably be encountering on this. So that's,
Yes.
if you have literatures that will say that this has been, already having this, how much percentage of acceptability or rejection, then, going forward with that is really gonna be good, I think. So that's one key thing that you have to kind of supplement in your proposal, that, yeah, this is having a very good acceptability. The other thing that you also probably would have to look into is the percentage of detection when a cancer starts. So I know that, so in the case of, say, dementia, you mentioned about dementia, Parkinson's, which is not cancer, but, some kind of syndromes or disorders, some of those is even the cells, our immune system itself, don't actually pick it up and give symptoms to the body until it's like really deteriorated. So like, for example, in your Parkinson's disease, that's why we don't have any symptom until like 50% of the brain is already damaged. And so that's when, how would you be able to get that early? What is early in terms of detecting the cancer? So, that might be one of the key things. You also would have to go and look for literatures and see, okay, if early detection means it's already this mass growth, then that's kind of, yeah, it fits the purpose. Does that make sense?
That's where it might be really good as a non-invasive, constant monitoring blood sugar.
Right.
It's just something.
Yeah, that's non-invasive. Yeah, like if you have something that's just here, prick it and see. That's probably the best fit for that
Yeah.
part. Okay.
Okay.
Very good. Very. Thank you guys. Thank you, Dr. Conlon. Let's give Dr. Conlon